to our second showing of works in progress. It's been a project through a class that we've started this quarter with 5 remarkably flexible and creative directors and a wonderful group of young undergrad actors as our acting company. The point of this class is to look at how we make theater, and maybe not plays, but theater experiences from a lot of different kinds of sources. We did a math project. We looked at the Syrian refugee crisis. We did another project on, well, we're doing a ritual project, which will culminate our week this week, as well as this project. This project really was born by a conversation with Michael Dennin that I had this summer about the idea of the intersection of science and the arts and how there are so many similarities in something he had seen. And I said, well, let's try it. Let's try a pilot of it. So I really want to thank the 3 scientists who opened their labs and their community to our directors for a very quick, a very fast conversation and take in order to start building some kind of theatrical expression. So I'd like to say welcome and welcome to the Science Fair.
Ha.
Let's take a look at pattern formations.
We've all been in traffic. When you're cruising along, and then all of a sudden, it comes to a stop.
Oh, come on!
And then it clears for no apparent reason. This is a great way to look at the things that we study. Why do things flow? And why do they not?
Bubble. Bubble. Bubble.
Bubble.
We generate bubbles! When we drag bubbles across a surface, the most common thing that they want to do is make the line with 2 bubbles on the side. They form a single file and then zip up 2 by 2. Occasionally, there's a mistake. The bubbles can angle, go out by itself, or break off. And if you don't pull it at all, you get a cluster of bubbles. What's kind of amazing about this is that it doesn't fold up or bend on itself.
Bubble.
Surface tension. It's called the Cheerio effect. If you pour Cheerios in milk, they come together and stick. Spontaneous symmetry breaking!
A non-linear way to look at the world. Say you take a pan of hot oil, and you heat it up a little bit, and you heat it up a little bit, all you'll get is slightly warmer oil. But there comes a point where if you heat it up a little bit, the oil will start to convect and form rolls, and a pattern of hexagons emerge. A small change caused a big change to happen.
Hi, I'm Michael Dennin. Yes, I'm the Dean of Undergraduate Education. Yes, I'm the Vice Provost. But I'm also a scientist. Over in my lab at the science-y part of campus, we look at all these things.
Spontaneous symmetry breaking.
Pattern formation. we look at soft, condensed matter, or solids that are squishy.
Like bubbles or foam.
These things behave like solids, but if you were to look at individual molecules, you would see that they have liquid attributes.
So foam, to find out if it's a liquid or a solid, really the ultimate experiment needs to be on the outside of the system of foam.
If you were to think about the way a system functions on a much grander scale, such as the nature of the universe, how much would you know about the nature of the universe if all of our experiments were to, by definition, happen in the universe? When Juliana and Paul came to interview me, they asked me why I do all of this. I'd love to share that with you. I study science because I want to understand the world better, and I want to be able to share that understanding with others.
Martin, and I'm a biophysical chemist. My research is focused on using the tools of chemistry to solve interesting problems in biology.
Hi, I'm Do Marine, and I'm one of her chemical biology graduate students.
I'm really interested in proteins. Ta-da! Bing. Ding. Wah. Ding. Wah. Ding.
And I make these proteins to study their structure and see what different things happen to them when you stick different chemicals in them. Like a chemical that causes them to unfold.
Now, I'm especially interested in the aggregation properties of proteins. Aggregation is what happens when proteins stick together in a way that's not natural or consistent with the functioning of the protein. Aggregations can cause diseases, such as Alzheimer's, Parkinson's, type 2 diabetes, and cataracts. Cataracts is what a lot of my research is focused on. Now, did you know that proteins make up the lens of your eye? Isn't that amazing? It's able to refract light because there's all these proteins stuck up in there in high concentrations. Now, the protein turnover is almost non-existent, so that basically means that the protein in your lens that you're born with are the ones you're going to have.
It is really, really hard for these proteins to stay stable and soluble your whole life. So if they start to aggregate in a certain way, that can scatter light, and that aggregate, that is a cataract. And we use box jellyfish lenses to do these experiments, and we use a gigantic magnet to do all these studies.
Now, we happen to study cataracts, but hopefully we'll be able to take some of the information learned from this research and apply it to the more complicated diseases I mentioned.
Gather round, children, it's story time!
Which one? Ooh, let's hear about the little engine that could.
Last week.
Okay, what about the one with the pigs and the wolves?
Okay, everyone. This is a story that you haven't heard. It's a story about mystery and discovery and a carnivorous plant.
What?
A carnivorous plant. That means a plant that gets most of its nutrients from eating small animals, such as insects.
I'm a vegetarian.
So do you want to hear it?
Yeah!
Okay. Once upon a time, there was a little girl named Rachel.
That's funny!
Who grew up in a place called Phoenix, Arizona.
Where's that?
Has anyone been to Arizona?
I know. My family went there last year.
Will you tell us about it?
It was hot and dry, and there are all sorts of pokey plants and weird animals and strange insects.
Well, Rachel was a very curious girl who happened to love all of these strange and exciting plants and critters that live nearby. She spent her free time catching snakes, scorpions, lizards, all to play with and observe. Like I said, she was a very curious girl and wanted to understand these strange and interesting things around her. Rachel's favorite subject in school was science. And after finishing high school, she went on to more school in hopes of becoming a real chemist.
When are we going to hear about the bug-eating plant?
I'm getting there. So with lots of hard work and lots of school, Rachel did become a real chemist. She also fell in love and got married. She got married to a man who was a statistician.
A what?
Who loved numbers as much as she loved chemistry. They would combine their skills of math and science to explore and observe and try to understand how things work, just like Rachel did when she was a little girl. Whoa. One Christmas break, Rachel and her husband decided to figure out the genome sequencing of a carnivorous plant.
Genome what?
Genome sequencing. It's this really exciting process of predicting what an organism's DNA is going to look like.
I don't get it.
You're gonna have to go to the library to read more about it.
I like libraries.
Now, during their research, Rachel and her husband discovered something very special.
What?
First, I have to tell you about a very famous man named Darwin.
Was he old?
In 1875.
Wow.
He wrote a book about carnivorous plants. He believed that plants had special things inside of them that helped them digest food, just like you or I. In all this time since Darwin wrote his book, no one had ever found anything like this in carnivorous plants. No one, that is, until Rachel and her husband. Wow. They discovered special digestive enzymes that in a carnivorous plant. Now, not only did they discover this enzyme, they were also able to use fancy technology to predict what these enzymes looked like.
Show me!
Me too!
Working with scientists to create an applied theater piece.
You're going to be working with scientists to create an applied theater piece.
That everyone gets a partner.
But Travis, you're flying solo.
And that we had to email our scientists ASAP to set up a meeting.
Well, go ahead, email him.
So at this point, I'm thinking, how am I going to make science theatrical? It's definitely not the most jazzed I've ever been in my life. A play about beakers and labs and white coats.
It's gonna be terrific.
So I emailed Greg. Dear Greg, I hope this email finds you well. First, I would like to thank you for agreeing to help me with my directing project. I would love to come in and meet with you and possibly see your lab if you are open to that. Here are some dates and times I'm available, blah, blah, blah. Warmly, Travis. And I Googled him. The Weiss Lab at UCI. Gregory A. Weiss at UC Irvine Faculty Profile System. UCI chemist finds a way to unboil eggs. No shit. Dear Jane, he's the unboiling an egg guy. I got his response moments later.
Dear Travis, We have our meetings on Monday at 4 and other meetings after that until late. You're welcome to come by and observe then. I've attached an article that I think might be pertinent to our discussion.
The article was about uncertainty in science and society, entitled The Scientific Method, Tales of the Unexpected, by renowned neuroscientist Stuart Firestein. I googled him too.
For scientists, uncertainty is the norm. Experiments begin with uncertainty. Why else do them? And even when they are successful, the results contain only a range of certainty and a range of confidence about that certainty. Yet, in the world outside the laboratory, uncertainty is perceived as negative. Billions of dollars of public and private money are poured into research.
Say that again?
Billions of dollars of public and private money are poured into research, largely on the false assumption that science provides cold, hard, immutable facts. This attitude is reinforced by an educational system that treats science as an immense fact tract, to be memorized and then largely forgotten, producing a populace that believes science is about answers rather than questions.
Now, for example, think about how many times you've seen coffee in the news. Studies say, it's bad for you, it's good for you, it wards off cancer and help grow your hair back. And all of this research, it might be perfectly valid, true, and scientific, but people don't understand the back and forth. They're disconcerted by the constant change. We scientists are used to that.
The real cunning of uncertainty lies in how it increases in every attempt to reduce it.
So, fast forward a few days to the Weiss Lab. Yep, they named it after him. Where Greg and his team of postdoc and PhD students are having a very important meeting about the progress of their current lab project.
Happy birthday to you, happy birthday to you, happy birthday dear Josh, happy birthday to you.
Pause. Okay, so you're probably thinking, oh, how nice, they got him a cake from Albertsons or whatever. No. There were fancy cheeses and crackers and a hummus platter. 4 different kinds of lemonade, a 300-piece plastic silverware utensil set. I momentarily considered a career change. So at this meeting, this girl was giving a presentation about zipping and unzipping enzymes. And she showed us an image of this data that had been kind of skewed, but Greg spinned it in this incredible way.
Oh wait, go back a slide. All right, you know what I really love about this? I mean, you can all imagine how scary it must have been getting this data, right? I mean, who in this room hasn't been here? It doesn't fit the model. But then they take it and you put it into some semblance of reality. I mean, initially, everything looks like garbage, but then they take it and they make it into something that makes sense and tells us something about the universe. That's heroic.
This is the kind of guy we're dealing with. Greg is a huge dreamer, a big ideas man. He went on.
Now, what we do has inherent risk, and my biggest concern is your personal safety. We can minimize that risk by being meticulous. This is extremely important to me, that nobody gets hurt. It's inherent in what we do. It's dangerous. All right, so for next time, I want you all to read an article on stress during your PhD.
It's called There's an Awful Cost to Getting a PhD.
I know how stressful it can be. I did it myself, but the thing is, you have to understand that you are not alone in experiencing this stress. We have resources to help you.
Next was a smaller group meeting with what I've dubbed the Eggheads. This smaller group was focused on the VFD,
Vector Fluid Device.
and unboiling an egg. Now, at this point, you're probably wondering, how do they do it? How do they unboil an egg? Well,
It starts with the principle of microscopic reversibility.
I didn't know what that was either.
Anything that can happen can theoretically unhappen if you retrace your steps.
First, we separate the egg white from the yolk. Then, we boil the egg white at 90 degrees for 20 minutes.
That's 90 degrees Celsius.
And we put the egg white in a water and a chemical called
Urea, a small molecule
Then we put the solution in a big glass tube and we spin it ridiculously fast,
5,000 rotations a minute.
making the solution spread into a thin film.
The proteins then stretch and contract until they snap back into the native shape.
This is called protein refolding.
The egg whites are back into its natural liquid state.
Ta-da! Pretty impressive, huh?
But I'm not so interested in uncooking your breakfast foods. I like my eggs hard-boiled. But the scientific applications of this go far beyond uncooking your breakfast foods. What we do in our lab is we use the vector fluid device to get proteins that are associated with cancer. Now, these proteins are at the helm of the cell. They drive the cell and they tell the cell, go cancerous, make tumors, spread around the body. These are evil proteins. But there's a really strong personal motivation that inspires me in this research as well. This affects my life. This affects the life of everybody that I know. And on a personal level, my father died of cancer at a ridiculously young age.
Our lab is focused on detecting cancer and detecting cancer early.
So that it will make it easier for the patients.
Makes it easier for physicians to be successful.
And it'll cost less, so everyone wins.
The unboiling an egg experiment relies on 10 different variables that all function independently from one another, making guaranteeing success tricky. And recently, the team has been unable to reproduce the same results. Frustrations have been high, and some have been considering walking off the project. Actual science isn't the only stressful part.
In reality, my contribution to the grand scheme of this project is like a grain of rice in a bucket. It's very minor, and it's disheartening. I came from biology from an altruistic standpoint, thinking, I want to help people, but I don't want to be a doctor. And then I find out that it takes 15 years for a drug to even be made, and $15 billion to accomplish what I want to accomplish. And that PhD that you've been spending 5 years on, I've spent my entire PhD on one protein that binds to another protein, just figuring out how that works. That's my entire 5 years. The beauty of collaboration is you don't have to know everything. But it's difficult sometimes when you don't speak the same language as the other scientists in your lab. There can easily be conflicts.
If we knew the stuff, we wouldn't be doing the experiment. I want the truth, but that can be a lot harder to get than facts. I can know that a series of facts are dependent upon a pile of variables that shift like sand on a beach. So I tell my students, give me the facts, but also give me the facts that don't fit. Give me the whole picture. I want the whole picture of the beach, not just one specific case of the individual fact on the sand formations according to this day and this time. Students and the public, they want science that delivers absolute certainty, solid fact, but scientific discovery doesn't work that way. Scientists are like explorers, and their mission is to enter the unknown, enter the abyss of the uncertain.
Scientists are like explorers. If we knew the stuff, we wouldn't be doing the experiments.